Moving on now to the second talk. As sort of we're now moving into I guess slightly smaller stuff. We're now going to be talking about particulate organic carbon in the deep ocean. So I'm going to hand over to Marty. Marty, it's all yours.
Hi, good afternoon, everyone. Thanks, Adrian. And well, thanks for having me today. And as Adrian said, I'm going to talk about the role of deep convection in the North Atlantic in injecting particulate organic carbon to the deep ocean. So as you can see, this is part of a team effort that lasted several years, in fact. And well, I am a researcher at the Marine Science Institute in Barcelona, but I started this work when I was at the Barcelona Supercomputing Center. Okay, so when we talk about the vertical export of particulate organic carbon, we know that the most studied and the most quantitatively most important mechanism is gravitational sinking, but carbon is also exported, particulate carbon, through other processes involved, including advective transports, the mixed layer PAM, so turbulent diffusive mixing, that is the one we will focus on today, and also active transport by mesopelagic migrants, among others. So focusing on the mixed layer PAM mechanism, as you can see in this cartoon from Leo Lacour, it's simply the vertical homogenization of the water column by mixing. So basically you see on the left an initial profile of POC, biomass, whatever. Then there is a deepening of the mix layer that redistributes this material vertically, so it depletes the surface and it inputs material to the lower part of the mix layer and that has been mixed, recently mixed. Okay, so this mechanism had been known for several decades. It's nothing new, in fact, but it had been studied mostly as a shallow process, and there was no global quantification that I am aware of until this work by Giorgio Dall'Olmo in 2016, where they used satellite data and biogeochemical algo data and also a climatology of the mixed layer depth to show these patterns that you can see here. So on the left, you see the deepest annual mixing, and on the right, the vertical fluxes due to the mixed layer pump. And as you can see, there's a strong spatial correlation between both. And an important conclusion of that work was that at high, well, at temperate and subpolar latitudes, this mechanism supplies an additional 23%, more or less, of POC to the mesopelagic layer, in addition to the gravitational sinking. So it's a relevant supply mechanism in regions where mixing is deep enough in winter. Okay, but these are climatological patterns, and we know that the convection mechanism it can be highly variable over interannual scales, and particularly so in the North Atlantic, as you can see in this graphic here from the work of Igor and Vyasheyev. So here you can see over 20 years of data in the Labrador Sea, and this is temperature data between the surface and 2000 meters, and these horizontal lines show the deepest annual mixing each year. So the different classes of Labrador seawater that were formed during different periods are marked here. And you can see that convection is intensified during multi-annual periods. And the last such period was between 2014 and 2018, more or less. Well, depends how you look at it. Here it says 2012 to 2018. And this was a very intense convection period that formed the most extensive modern Labrador seawater class since the early 90s. So important event. And this is the period I'm going to talk about because, lucky enough, there was lots of biogeochemical Argo observations during that period in the subpolar North Atlantic. So here on the top right corner, you can see the trajectories of several Argo floats that were deployed in this region in 2013, 2014, or by the team of Hervé Clostre in Villefranche, in France. And this is an amazing data set. It has been extensively used in many publications. And it's a data set I fell in love immediately when I started to work on this data almost 8 years back now. And important, Argo floats are taking, as you know, regularly vertical profiles. So when they profile from the parking depth at 1,000 meters up to the surface, now they collect measurements, they transmit, they dive again back to the 1,000 meters parking depth. But Argo floats are also taking measurements while drifting at 1,000 meters. And in this particular data set, these floats were programmed to acquire bioptical measurements every 10 minutes. This means over 140 measurements per day when there is no vertical profile. So here I'm combining these data sets to look at this process, the mixed layer PAM or convective mixing of particles in winter, spring in this area. So let's start looking just at 1 year of data from just one float, which is particularly nice. And this float was in the Central Labrador Sea. Here you can see on top the sequence of vertical profiles of chlorophyll fluorescence. And these lines, and the dashed line, for example, is the mixed depth according to a very fine density criterion, 0.005 kilograms per cubic meter, density difference between the surface and 1,000 meters. So this is probably the very actively mixing layer. And you can see that as this layer deepens, the chlorophyll profile is homogenized, but very interestingly, we see a jump in chlorophyll fluorescence at 1,000 meters, right at the moment where this deepening reaches 1,000 meters, of course. And chlorophyll fluorescence at 1,000 meters reaches above the background noise levels and reaches relatively high signals comparable to the oligotrophic surface ocean, for example. And this signal persists for as long as convection is deeper than 1,000 meters. And this allows, but this together with the vertical profiles allows us to segment this time series into the pre-convection period in yellow, the convection in blue, and the post-convection in green, which I will use then to derive some statistics. We see the signal in chlorophyll fluorescence, but also in backscattering, although more modest. And we can also clearly see the signal of convection in these stairs in temperature. And we can see successive convection events matching successive peaks in chlorophyll fluorescence. Sometimes, not always. This is the largest event that I used to illustrate this process. So that's clear. Convection is bringing these surface particles to depth, deeper than 1,000 meters. And if you look at the entire time series from this float that I'm showing here, and this is very nice because this float drifted for over 4 years in the region, you can see that more than one event was recorded in successive winters. So here there is a bit too much of information, but on top you see the chlorophyll fluorescence, bulk signal, the spike signals, which belongs to large aggregates that develop during the bloom and post-bloom seasons, not during the convection season. Below in brownish colors, you see the backscattering signal, bulk and spike. So again, large aggregates. And here below, you see the entire trace of chlorophyll fluorescence at 1,000 meters, backscattering, and temperature. So, and this box is highlighting the year I was showing before. So you can see here several events with a coherence between the 1,000 meter and signal, the profiles. There's also synoptic coherence with satellites, which sometimes when there's observations, which are scarce, which I'm not showing today. Okay, so we could confidently detect 12 of these events, which are shown here on the map on the right. And they are shown together with the contours showing the deepest annual mixing in the region. So you see that the events occur, obviously, within the deeply mixed region enclosed by the contours. And here you see the temporal trace of chlorophyll fluorescence with a removed background and plotted along the day of the year. And as you can see, the duration of the events is very variable, can be, we detected up to 80 days of duration. And the magnitude as quantified by the peak chlorophyll fluorescence is also very variable. So the event I was showing was the largest one here with this chlorophyll fluorescence signal. Okay, so now keep this in mind, but before I go into the next part, I would like to explain also what bioptical data tell us about the properties of these particles. And by this, we are using the vertical profiles. And the first metric I'm showing here is the equivalent spherical diameter, which is based on the variance of the backscattering signal. And you see profiles in different colors. So yellow is pre-convection, and blue and purple are convection, and green is post-convection. And you see that at least proxy for particle size shows much larger sizes during the post-convection season when large aggregates are exported after the bloom, right? But during convection, the particles are the smallest. And if we distinguish between active convection with the weakest stratification and arrested convection, so in this intermittency between convection events, this is the purple we see, also see slight differences. So injected particles are small, and they are also fresher, much fresher than the particles that we typically find in the mesopelagic layer and below. And here we quantify this with this proxy, which is the fluorescence over backscattering signal, which has been used as a proxy for specific POC content, hence freshness or lability of the particles. So small, fresh, labile particles injected to the deep mesopelagic and below. Okay. Based on the peak fluorescence signal, we estimated the equivalent POC, particulate organic carbon, phytoplanktonic POC, sorry, using estimates of the carbon to chlorophyll ratio. And given the several sources of uncertainty, we are giving a range of estimates and a central estimate based on the previous light exposure based on satellite and Argo data. So the conclusion here is that this increase in POC approximately doubles the background POC before the convection. So it's an important input. And this metric is also very useful because it allows us to compare to model simulations, which is the second part of the talk. Okay, so to try to make some sense or to get some mechanistic insights on these observations, we used simulations with the NEMO4 and Piscus model. And you see here a cartoon of the model with two phytoplankton groups, two zooplankton, two classes of sinking detritus that suffer lots of transformations from detritivory transformations, fragmentation, and so on, decay to DOC, and eventually remineralization to DIC. So a first simulation I'm going to show corresponds to the exact same place of the large event I was showing at the beginning for the year 2015. And here you can see the small POC, which is the sum of phytoplankton and small zooplankton and small detritus in the vertical profiles, so analogous to what we plotted from Argo floats. And below you see the temporal time series at 1000 meters of the total POC, and it's broken down into the different components. So phytoplankton, small detritus, large detritus, and micro and mesozooplankton. So first important thing, sudden increase in the POC as soon as the turbulent layer reaches 1000 meters. Looks like the observations more or less. And second important thing, variable contribution of phytoplankton to this increase. This is just one event, but this fraction can be very variable. And we also see an increase in large aggregates in midsummer, matching also quite well what we saw from the spike signal in bioptical observations from Argo floats. So qualitatively, this resembles quite well the observations. But we can go more quantitative than this. And to do so, we'll use this delta POC and delta POC phytoplankton, no, phytoplanktonic carbon increase. So first, before I do this, I'm showing here that after this one, the simulations, we run 3D coupled simulations, so with online biochemistry. And these simulations were run globally for, well, following this OMI protocol using atmospheric forcing, so that the ocean physics are aligned with atmospheric forcing. And this reference simulation covers the period between 1958 and 2019. We ran the simulation, but then, as I'm showing here, we noticed some biases in this 3D simulation, the default one. And in particular, we observed too vigorous mixing during some years, especially during strong convection years, which led to excessive inhibition of phytoplankton growth and excessive depletion of surface biomass, as we can see when comparing the simulation to satellite observations of phytoplankton at the surface. Here you can see, especially in the Labrador Sea, that during this 2014 to 2018 strong convection period, there is stronger negative bias, especially in the Labrador Sea. This is due to structural model biases, probably. And we thought of a strategy to kind of short-circuit these limitations of the model, which was nudging the model toward satellite observations of phytoplankton at POC. So adapting the protocol that is typically used for physical tracers, so salinity, for example, we applied a similar approach. We tuned it for phytoplankton biomass, so it follows more closely observations. So this is our second 3D simulation, which is called SAT and covers the satellite period. So it's interesting to see when we compare this delta POC, so the peak excess POC added by convection, right? When we compare this between the observations and the simulations on the right, that the ref simulation looks to underestimate this, but with the satellite nudging, this metric gets closer to observations. And of course, this process has limitations, but it's an interesting observation. So we are going to use both simulations in parallel. So can we talk now about fluxes, not just more descriptive metrics? So what is nice about biochemical models, which have many limitations, is that they allow us to extract biochemical fluxes and also physical transports from a coherent dynamical framework, right? So this is what we did with our model, extracting different types of POC transport during the simulation. And I'm showing this using the entire reference simulation to begin with, as you can see here. So between 1958 to 2019. So on top, you see the NAO Winter North Atlantic Oscillation Index, which you probably know is the main atmospheric mode that forces these interannual changes in convection in the Labrador and Irminger seas in the North Atlantic. Below, you see another metric, which is the deep convection area, where annual convection exceeds 1,000 meters, and you see that it has these cycles of intensified convection in the 70s, the most important one in the early 90s, and this recent one 1 decade ago. And we can also see that there is a strong correlation when we low pass filter the data using a 5-year running mean. So what about the fluxes? Here below you see the gravitational, diffusive, and advective fluxes. So diffusive fluxes include the fluxes due to convection, so overturning instability, and also the background diffusive fluxes, which are weaker. So we are pulling all of them together here. So you can see that while the gravitational fluxes have some oscillations but are more stable over interval scales according to the model, the diffusive fluxes driven by convection follow these cycles of convection, of course. And regionally, they can account for a substantial fraction of the vertical POC transports at 1000 meters. I'm showing also advective fluxes, but this is something I'm not going to talk much about today. Okay, so how does this look spatially? Now on the right, you see the contribution of diffusion to the total diffusive and gravitational fluxes at 1000 meters during 2 periods. So relatively weak convection period between 2009 and 2013. And below you see the contribution during the intensified convection period when this mechanism was supplying an additional 30, 40, 50, 70%, depending on the place, POC in addition to gravitational fluxes. Okay, so it's very interesting to look at fluxes at a specific depth. It's a very useful metric, and easy to compute, an easy one to compute. But when we think of carbon sequestration or mechanisms contributing to longer-term carbon sequestration, it's more important to look at volumetric budgets, right? So here we decided to look at the net diffusive supply between 500 meters and 2000 meters. Why this depth? Because every winter mixing is going as deep as 500 meters in this region. This means that any inputs to this layer on a given winter will probably be mixed again and exchanged with the atmosphere on the following winter. Whereas mixing between 500 and 2000 meters is more likely to stay for a longer time in the ocean. And to quantify this, we compared the deep convection volume. So and you see there is this strong relationship. And you see also that the relationship is slightly different depending on whether we compare the reference simulation or the satellite nudge simulation, but overall the range of variation is not so huge, which allows us to give a range for the estimates of convective injection of particles to the deep North Atlantic subpolar gyre. And finally, I'm going to conclude now. We also computed the total POC budgets and found that this enhanced supply during periods of deep convection was translating into enhanced degradation rates and remineralization. And this is super interesting because we found also some observational evidence for this enhanced particle degradation rates during this recent intensified convection period. And very interestingly, there is also an enhanced lateral transport away from the convection region during periods of intensified convection, meaning that part of the particles that are injected vertically then are spread horizontally through mostly advection or lateral diffusion, which in the model, which has very good resolution, is accounting for unresolved scales of advection, right? And this is it for today. So these are the take-home messages. We saw, using biogeochemical flows, we saw these recurrent chlorophyll fluorescence peaks at 1000 meters, which were kind of unexpected. In the deep convection region of the Labrador and Irminger Seas, these injected particles are rich in phytoplankton material, so they are relatively small, reflecting the properties of phytoplankton in winter in the area, and fresh compared to the typically older detritus that are found suspended in these mesopelagic or bathypelagic depths. We found that we can reproduce some key features using a biochemical model, and using this model to quantify the fluxes, we find an important interannual variability and signal of this, and an important variability of these convective inputs to the deep North Atlantic. So Basically, the diffusive POC supply approximately doubles between years of weak versus strong convection in this area. And we hypothesize that these injections reshape deep ocean carbon budgets because they can trigger different types of bacterial metabolism or sustain zooplanktonic or metazoan populations and helping them survive before the aggregate the gravitational inputs that happen later in the summer. And also this drives lateral exports. So that's it. Thank you very much for listening, and I will be happy to take questions. And here, if you can scan this QR code, you can download the preprint where all of this is explained probably much better. Thank you.
Excellent. Thank you, Marty. Another excellent talk. Anybody have any questions they'd like to ask? Either either put it into the chat or just sort of open your microphone. I should check the chat actually. So I've got one, Marty.
Yes.
I guess one of the difficulties in comparing biological carbon fluxes is how you choose the depth to do it at. And so the gravitational flux is usually largest at the surface, but it very rapidly attenuates with depth. So by the time you get to 1,000 meters, then you're down to, the order of 10% or less. Whereas presumably this convection flux you're talking about is much more efficient because you're transporting material before it can degrade. So when you were talking about the comparison to the gravitational flux, was that based on a surface export or was that based on the material making it to the same depth of around 1,000 meters?
Thanks for the question, Adrian. So it's good to clarify this, and I'm comparing the fluxes at 1,000 meters.
Right.
So diffusive and gravitational. And of course, the comparison is only with model data. So there is almost no measurements at 1,000 meters in this particular region and periods that we can compare to. In another paper, we have compared in the North Atlantic, the whole North Atlantic, the gravitational fluxes from surface to bathypelagic depths between these same simulations and data sets like from sediment traps and thorium. And they compare reasonably well with a lot of scatter, but suggest that the model is doing a decent job at reproducing the fluxes at 1,000 in the lower mesopelagic. But of course, there's always a lot of uncertainty in this kind of estimates.
Yeah, sure, of course. No, thank you.
But yes, I agree that this turbulent transport of particles is very efficient because basically it's transporting in a matter of hours or a few days that fresh material from surface to beyond 1,000 meters.
Yeah, I've got another question, but I should see if anybody else has got one before I ask it. Has anybody else got anything they'd like to ask? Okay, so one thing I've become interested in. So in your model, Pisces has an aggregation term, so that phytoplankton. So when you have deep convection, you have elevated turbulence. Do you actually get increased aggregation as a result in the model as well? So you're effectively exporting material which has additionally been aggregated.
Yes. Yes. But the way this is represented is very simplistic. So basically, in Pisces, there is an increased shear rate in the turbulent mixed layer, and this is driving the enhanced aggregation and in the mixed layer. So
Right.
basically, this mechanism is kicking in when the convection occurs. And something we show in the paper, in the preprint, sorry, and I couldn't show today, is that these turbulent inputs, then they increase gravitational export by large aggregates at 1,000 meters and beyond in the model through this mechanism, which is very interesting, and it matches some observations. No, I remember there's this paper by Sari Giring in 2016, and I think they were showing that some clues that the mixed layer PAM was supplementing like small particles that then could aggregate and supplement the gravitational fluxes. And it's interesting that this mechanism kind of occurs in the model, although
Yeah,
idealized, very idealized.
yeah, fascinating. The more people look at it, sort of the more these connections between these pumps appear. But that's fascinating. Thank you. We should draw
Thank
the
you.
call to an end. So thank you very much for the talk. Once again, encourage anybody who would like to ask Marty any questions after the talk, please just email him. But otherwise, thank you both to Marty and to Sven for two excellent talks, and I hopefully see a good audience on our next calls we'll be arranging in March and April. But otherwise, thank you all very much. Goodbye.